So I suspect that most of you were here yesterday for Dr. Call's session, as was I, hiding in the back. And I know that because of some questions, excellent dialogue back and forth, they didn't get through everything. So I've thrown a few slides in at the beginning just to make sure we're all starting from the same position. So this session is really to focus on clinical trials in mantle cell lymphoma. That's my name, Mitchell Smith. I recently moved to George Washington Cancer Center, and Dr. Sotomayor is there, who's helping run the trial, the session here. Dr. Dunleavy is there. So we have pulled together a pretty impressive lymphoma team, if we don't say so ourselves. And my job is to oversee clinical trials in the cancer center over all of things, not just lymphoma. So it's my job to get clinical trials up and running there a little more robustly, but clearly we have an interest in building our lymphoma clinical trial program. So today I'm just going to spend a few minutes reviewing current status of treatment in mantle cell lymphoma. Then we're going to talk about so-called novel, or say not so novel approaches. Ibrutinib's been around for 4 years now. Show you what current trials, some of which have actually finished accruing, but we are waiting for the results, and we have to plan our other trials without knowing those. And then what's ongoing right now in clinical trials, and the kinds of things that, if you've had previous treatment and are looking for trials, the kinds of things that are out there currently and on the horizon. So the challenges, I was thinking about MCL, mantle cell lymphoma. It's certainly not the most common lymphoma, so we call it the most challenging lymphoma. And as we heard yesterday, we don't think it's curable today. If I did have a treatment that cured it, I wouldn't know that for many years. So some people might be cured with current treatment. We just don't know it. We have to wait and see. As you heard yesterday, the MIPI, the Mantle Cell International Prognostic Index, and proliferation, usually measured by Ki-67 staining or fancy gene expression profiling, is the best we have to kind of say who's going to need treatment sooner, who's going to do better with treatment. But we really need better markers. All the genomics that we study today in certain diseases has said, oh, that's the key gene, but in mantle cell, when we do that, we just come up with proliferation. So whatever is driving your cells to divide faster makes it a harder disease to deal with. But in each patient, that may be different. There's not one target. As we get better, patients are in remission longer. So if we wait, and our endpoint of a trial is when do you relapse, the good news is that's further and further out, but it makes it hard to generate the next trial because you have to wait until you see which treatment was better. So now there's a lot of focus on interim endpoints. So if you achieve a complete remission at the end of treatment, is that a good thing? Well, yeah, but does it translate into longer, better outcomes? We don't know that. And how you define a remission. So, we talked yesterday in the clinical trial session, partial remission, 50% shrinkage, okay, fine. Complete remission, full, the CAT scan goes back to normal, no lymph nodes. These days, the CAT scan might not be totally normal, but the PET scan, which measures radioactive sugar uptake, positron emission tomography, the PET scan, that's a marker that hasn't really been validated in mantle cell. It's a really good marker in diffuse large B cell that we know you have to be negative at the end of treatment. We think that's a good thing in mantle cell. We're looking at it in some of the current trials, but we don't know that. And then that's one level, and then even at another level, this topic that's come up during the last 2 days, is the biological endpoint, minimal residual disease, by fancy tests in the blood and the bone marrow. Have we gotten rid of all the cells? And what level do we call it? We know that there's probably cells there because we didn't cure you, but is it 1 in 1,000, 1 in 10,000, 1 in a million? What level is really what we're targeting at? And does that then translate into better outcomes? So these are all things that we're looking at. They might be piggybacked onto a clinical trial that you're in, where at the end of treatment you're going to get a CAT scan, a PET scan, and blood work. Maybe the MRD levels will be followed over time. So that's sort of built into the trials. And we'll talk about, stem cell transplant. A lot of you have been recommended to have that or have had it, but there's biases for and against. Patient might say, I don't want to do that. Doctor may say, I don't think you're quite up to it. So biases may say, I don't want to go into a trial because I might be treated intensively, or I might not be treated intensively. We all have biases about what we think is the best approach. And ultimately, we'd like to tailor therapy for each patient. So I talked about the MIPI, and the MIPI-C is sort of combines the MIPI and the proliferative rate. It looks like 30% Ki-67, 30% proliferation seems to be a cutoff for tougher disease, more than 30%, better disease, less than 30%. There was a lot of talk in yesterday's session about P53 mutation, and the new paper that just came out, whether it's a mutation or a deletion may be different. But clearly we need better markers to really pin down prognostically who's going to do better and worse, and that helps us decide who needs intense treatment, who needs less intense treatment. And I'll show you a trial later. We're looking response adapted. So maybe we can say, all right, I don't know starting out how to define your disease, but if I give you a treatment and you've achieved minimal residual disease, you've had a great response, maybe then I can say, oh, you don't need to go to the next step with the stem cell. So we're asking that question. And the last thing is just to point out that, we all know that mantle cell is characterized by the 11-14 translocation, expression of cyclin D1, and that's a nice target, but cyclin D1 by itself doesn't cause mantle or result in mantle cell lymphoma. There are additional changes, such as P53 or ATM or these things that Dr. Call had on his slide yesterday. So, and it doesn't seem to be one gene or two genes in everyone, so it's a mix. What do I consider the most pressing questions in clinical trials today? So it's different if you've never been treated or if you're treated. So I think in newly diagnosed, if you're deciding what's the best treatment for me, you heard a lot earlier today about the first question, which I didn't put on there, is do I need treatment? So that's the first question. If I have no symptoms and this was found incidentally, maybe I don't need treatment right away and I can be observed. But then once you get to the point where you need treatment, you have symptoms, your blood counts are abnormal, what's the role of stem cell transplant in the era of newer treatments? Maybe we don't need it as much because we have better treatments. These biological agents, ibrutinib, venetoclax, these things that we're going to talk about, do we add them to chemotherapy, the backbone, to improve outcomes? Will that improve them? Do we add them with it? Do we add them after it? And ultimately, can we get rid of the chemo part and replace treatment with biologic therapy that might be less toxic and more efficacious? And since we don't cure you today and you get into remission with an induction or a transplant or whatever you do, what is the role of maintenance or consolidation? I use them kind of interchangeably. I think of consolidation as I've got a little disease left and I'm going to treat you for a period of time to try to knock that down. Maintenance I think of as well, really maintaining the remission as long as possible. So we talk about how long to treat rituximab. If you give it for a short time, I consider that consolidation. A long time, I consider it maintenance. But those are sort of vague terms, so no one really agrees on those. But, I think what do we do after we get you in remission to keep you there as long as possible? So those are what I think of as the questions that need to be asked in newly diagnosed. If you've been treated and the disease has come back, that's where the biologic agents are approved and are being studied. How do we add them back properly? Again, the same question, do we add them to chemotherapy? Do we replace them? We have all these options now. How do we decide? One drug, two drugs, three drugs, sequentially, all at the same time? These are questions that take a lot of finesse to kind of answer, and we can't ask every single question with the number of mantle cell patients we have. So we need to sort of strategically think about how to ask those questions. And right now, if you go on ibrutinib, you stay on it until it stops working, or you have side effects that mean you can't take it anymore. But, ultimately, in chronic lymphocytic leukemia, we use the drug similarly. People are starting to ask the question, can I stop? If I get that MRD level down where I can't detect it, maybe I can be off treatment. Maybe I don't need to be on ibrutinib the rest of my life. Maybe I can stop it and it'll work again in a year or two when the disease comes back. Maybe the disease won't come back. So again, interesting questions we need to ask. All right, so especially because Dr. Call didn't get through everything yesterday, I'm just going to have a couple slides here on what the current treatment is for mantle cell. So what we generally decide is if you're young and fit, and that is in the eye of the beholder, young for a physician is usually about 5 years older than me and younger. So it keeps raising up, and older and less fit. But again, if you're 60, there's 60-year-old marathon runners, and there's 60-year-olds who have had 3 heart attacks, and you can't really treat them the same. So someone who we think can tolerate intense therapy, I would say there's probably 2 main treatment options. There's this Nordic regimen, and then there's this regimen that was developed in Germany. There's 2 others I listed there. The hyper-CVAD is sort of falling out of favor because of some problems with going on to the stem cell transplant that may or may not be sort of correct, but some trials suggested that. So that's sort of fallen out of favor. So, but when you look at these, this is a CHOP-like regimen and high-dose Ara-C. This is CHOP and a high-dose Ara-C containing regimen with another drug. The hyper-CVAD is a mix of the same drugs as in CHOP with some Ara-C and methotrexate. So all of them are relatively similar. You get a CHOP-like regimen with rituximab, you get a high-dose Ara-C with rituximab, and you alternate that for some number of cycles, 4, 6, or 8. And then you go on to the autologous stem cell transplant. And now the question is whether you should get rituximab maintenance after that. But I think that is sort of the standard upfront therapy if you think you can have intensive therapy. And the thought is that that gives you the longest disease-free interval, but unfortunately patients continue to relapse over time. If you're not as fit or you say, I'm not going through that, then most of us would use bendamustine rituximab with rituximab maintenance, although there's some question about the role of maintenance. Our CHOP with our maintenance looks okay. Yes?
I'm sorry to interrupt. The one that's just above older fit, under young fit?
CALGB regimen?
I haven't heard of that.
Yeah, it's a really complicated regimen that they devised, and it's good, but it's very intensive, a lot of side effects, and I sort of put it on there for completeness, but I don't think anyone's using it outside of there were some clinical trials looking at it, and the results are okay, but it's not one that most of us use.
How long ago was that around?
The trial was published probably 4 or 5 years ago. Yeah. And, the trial that said our maintenance was good was actually after R-CHOP. So, R-CHOP alone we think is not adequate, but R-CHOP with our maintenance, those patients did okay. There's one replacing the vincristine, the O is oncovin or vincristine here, with Velcade or bortezomib. And that was a randomized trial without maintenance that the VR-CAP regimen was better than R-CHOP. There have been trials that say that bendamustine rituximab is better than R-CHOP. We don't know how bendamustine rituximab compares with that. And then Dr. Call yesterday showed the ECOG data where we had the CVAD, not with the high-dose Ara-C, but just the CHOP-like part of CVAD, adding bortezomib or Velcade to that with our maintenance, and the results look pretty promising. But all phase 2 trials, and again, patient selection might vary. So I would say those are sort of your choices, but most commonly I would say in this country, bendamustine rituximab with rituximab maintenance is sort of standard. This question about stem cell transplant, I just want to take a moment because we all accept it as standard. I'm not sure there's data to say that it should be used in everyone. So the patients who are on those stem cell transplants are young, healthy, and most of them have low or at most intermediate MIPI scores. So it all goes back to patient selection. There's no the randomized trial which suggested transplant was better in progression-free survival goes back prior to rituximab and all this other stuff. So it's not modern data. The patients we really want to help are the patients with the high-risk MIPI. They don't really do as well, so great after transplant. So it's like, well, I'm treating the people who don't need it, and they're doing well. The rituximab now, or even radioimmunotherapy, prolongs progression-free survival. So if you think of transplant as a consolidation treatment, maybe rituximab or other treatments like Revlimid rituximab might actually replace transplant. So now we're going to ask those questions. And the patients who have a good prognosis do well. So anyway, so it's accepted. We all do it. because we don't have data not to, but it's, things that are accepted aren't, we always have to decide there are certain things we do because there's great evidence, randomized trials, and we know this is what we should do. And there's other things we do because they make sense and we're comfortable with them and we've had good results with them, but it doesn't mean that they're written in stone. And we always have to think about those are good questions for clinical trials. I just show this. This was a trial that we did in Eastern Cooperative Oncology Group. The 99 means it was designed in 1999. It actually didn't open for 5 years, which is another long story. It was the first mantle cell trial done, specifically mantle cell trial done. It accrued very nicely in a little over a year. And it was a very simple trial. It was R-CHOP for 4 cycles, not even 6, followed by 1 dose of radioimmunotherapy, this drug yttrium-90 ibrutinib, better known as Zevalin, which doesn't get much mention, but it's a good drug. And that's a pretty simple treatment. It was designed for anyone, not transplant. Transplant wasn't used as much back then. And what I want to show you is that results is overall survival, and Dr. Call, and we mentioned we don't always like to show that stuff, but patients who are young with low intermediate MIPI, the kind who kind of go on the transplant studies, this is 10 years, and half of those patients are still alive at 10 years. So is it just that this is a great treatment, or is it just that we select patients who are destined to do well? And so when you look at trials, it's really important to look at who's in there. Are they patients like you, or are they patients who have different characteristics? All right, so when I say not so novel agents, we have in the United States 3 drugs approved specifically for mantle cell lymphoma, relapsed mantle cell. We have bortezomib, better known as Velcade. So that's called a proteasome inhibitor. It blocks protein degradation in the cell. The cell basically we think gets, there's certain genes which have to be degraded, and when we block that, the cell doesn't like it, and it dies. Lenalidomide, we don't really know how it works. It has to bind to a protein in the cell, but whether it affects the cell directly or the immune environment around it or both. But lenalidomide's been around for a long time for multiple myeloma and other diseases, but approved for mantle cell. And the new kid on the block, ibrutinib, which I suspect if you have mantle cell and you have relapsed after, you probably had that drug. In Europe, they approved ibrutinib. They did not feel that the evidence for bortezomib and lenalidomide were worth approving, but they approved another drug called temsirolimus, and this is a diagram. It hits this enzyme called mTOR, which is a signaling pathway in the cell, which you don't need to know. But these drugs have been around for a long time as well, but there was data that it has a reasonable response in mantle cell lymphoma. And so, but there was in Europe, they did a randomized trial of ibrutinib versus temsirolimus, and ibrutinib was the clear winner. So if you have a choice, ibrutinib was the winner. But, this drug we shouldn't throw away. It does have activity. It might be useful in combinations as we go forward. So this is one of those complicated, what keeps a cell alive? So B cells, we're learning a lot about the biology. Learning about the biology helps us figure out what the best treatments are, and it'll be printed somewhere and you can look at it. But the bottom line is that there are all these signals, complicated signals, that tell a cell to proliferate. And over here, there's a whole complicated system that tells a cell whether it should die or not. And we want to block proliferation, and we want to promote the mantle cell death. So in here, drugs which block, which affect the cell death and promote cell death are drugs like venetoclax. Up here, you've got ibrutinib. We'll talk about some similar drugs and a whole host of possibilities. Here's everolimus. This enzyme called SYK or SYK has a drug which inhibits that. So you can inhibit many steps here, and then you can combine them in various ways. But the idea is that there are lots of opportunities here for investigation, almost too many. How do we pick which ones we want to do? So we might say, this is all in the same pathway. I want one drug out of pathway A and one drug out of pathway B plus one drug that hits cell death. That might be a rational combination to try. So when I thought about what are the novel pathways, so I'm not going to go in order here. So we talked about cell death. Apoptosis is a fancy name for programmed cell death. There's venetoclax. There was an earlier form of venetoclax. Talked about the proteasome inhibition. I talked about bortezomib, but there's also carfilzomib and ixazomib. Again, drugs which are similar but somewhat different and might be worth trying. There's some literature on this old-time drug that inhibits DNA and RNA synthesis called cladribine that might be particularly active in mantle cell. We don't sort of throw it away because it's like a chemo drug and it's not as sexy as the new targeted ones, but there's some interesting data that it might work as an epigenetic modulator. We have the lenalidomide. We have a whole host of antibody-based. So we talked about cell signaling inside the cell, but on the surface of the cell are these targets. You've all had rituximab if you've been treated. It binds to CD20. What does that do? So it might make the cell die. It might make the immune system recognize the cell as having an antibody on it and kill it. So there's a number of ways, and people have spent 20 years since it was approved trying to get better rituximabs. We haven't always gotten that way, but there are some new ones that look promising. Gazyva or obinutuzumab looks good. You can have antibodies that target other things than CD20. CD19 and CD22 are on all sorts of B cells, so they are reasonable targets. CD22, so rituximab, when it binds CD20, stays on the surface, so the immune system can recognize it. CD22, when it binds, gets internalized, so that's a way to deliver a drug to the cell. So you put a drug on the CD22 antibody. The CD22 antibody binds to the cell. The cell says, oh, I'm bounded, I take it inside. Now you put the drug right inside the cell. It doesn't get into the other cells. That's called an antibody-drug conjugate. The famous one is brentuximab, used in Hodgkin lymphoma and some other lymphomas. But there are CD22 drug conjugates, CD79, a whole bunch of antibody-drug conjugates in clinical trials. They're pretty nice drugs. They don't really have a lot of side effects other than from a little bit of the drug that leaches off. Most times the current drugs they use is neuropathy, so those are good. And then this idea of bispecific antibodies I'll come back to when we talk about immune therapy. And then this one, cyclin D1. That's 11-14 translocation. Why haven't we targeted cyclin D1? Well, we haven't been able to. It's not for lack of trying. But it turns out that cyclin D1 is phosphorylated by the cyclin-dependent kinase. And so now there's inhibitors of CDK, and there's this drug called palbociclib that we did studies on a number of years ago. This drug is now approved in breast cancer. Turns out to be a remarkable drug. There's actually 3 CDK inhibitors now for breast cancer, which is good news for lymphoma doctors and patients, because if it was just a lymphoma drug, it might not make it. But a breast cancer drug, boy, they're going to market that one. So again, this I think is an exciting area that it is being explored. So there's lots of new things, and we just have to figure out how best to get there. So I want to talk now about the trials that are basically accrued. There's a couple of main trials accrued and a couple that are working there that are open, and I'm going to talk about those, and then we'll come back to what new ones. So this is a trial that ECOG ran. It is called 1411, which means it was open and designed in 2011. And it sort of uses the BR followed by rituximab as a standard, even though it's not always a standard because people argue about rituximab. And it asks whether adding Velcade or bortezomib to induction, or lenalidomide to rituximab during the maintenance, or both the Velcade and the lenalidomide, which of those is the best strategy? And about roughly 80 some odd patients in each of those 4 arms. The last patient enrolled on the trial last September. That meant they finished their 6 months of induction therapy in March. We have to follow them long enough to see how things are going, but we hope at the big ASH meeting a year from now, so December of next year, to be able to present at least the early results. Obviously, they'll have to be followed longer. But remarkable that a U.S. national trial could, in about 3.5 years, accrue 370 some odd mantle cell patients, which I think is remarkable. It was open for technical reasons, it was really designed for over age 60 non-transplant. We opened it because the transplant study closed to anyone, but only a few patients under age 60 went on because there's still this bias to transplant, which is fine. So it's basically a trial for patients over the age of 60, and hopefully in a year or so we'll be able to tell you the results. With this trial, we looked at, in all these patients at the end of induction, CAT scan, PET scan, MRD by biology, by DNA sequencing methods in the blood and the bone marrow. So we will hopefully, from this trial, be able to tell you which is the best end of induction treatment, a marker of response, and, does MRD predict how long you're going to stay in remission? So lots of biological things. There were quality of life studies. So patients on the trial are taking questionnaires every 3 months to tell us, did they have neuropathy? Did they lose their hair? Did they have infections? So that we can sort of compare, if there isn't a big difference in some of these arms, at least we can declare a winner based on side effects. Yep.
Why do you think you got such a huge response in the participants?
Yeah, so the question is, why did we accrue so well? I think people with mantle cell lymphoma, because they're doctors and patients, because there is no accepted therapy, are very willing to put patients on protocol. If you have an accepted therapy, it's a little bit hard to say, I'm going to get something different. But here, it wasn't a radical design change. Everyone got the standard treatment plus, so it wasn't like, gee, I'm not sure, I'm not going to get chemo, maybe that's bad. So it looks complicated with the 4 arms, but it's basically an easy study to understand for the doctor and the patient. It builds on standard therapy, and people are very willing to enter trials with mantle cell lymphoma because we don't have a good answer. So it's all of those things together. I'll tell you in a minute why we don't do some things, because we don't think we can accrue in certain situations. But yeah, so I think, and we're pleased with that, and that gives us confidence that we can design other studies similarly and keep the momentum going for clinical trials. So this was the companion trial. So at the same time we opened that trial through Eastern Cooperative Oncology Group, but with all 3 national groups participating, and eventually the National Cancer Institute of Canada actually joined it, there was another trial for younger patients who were destined for stem cell transplant. And the question was, a lot of people used 4 cycles of the R-hyper-CVAD full regimen and then transplant. And the question was, did you really need to do that? Or if you're going to the intensive therapy with transplant, could you do well just with bendamustine rituximab? There was a lot of bias here. Some people said, I'm not putting my patient on bendamustine rituximab. That's not intense. Other people say, I'm not putting my patient on hyper-CVAD. That's too intense. So, there were some problems. But the interesting thing was this is an example of where the process kind of works, but it wasn't the answer we wanted. There was a built-in concern really for the bendamustine that maybe that would knock down your counts and you couldn't collect stem cells and get to the transplant. It turned out it was the other arm, the hyper-CVAD arm, that had the problem, but there were designed things in there. So very early on, 20 patients on each arm, there were a certain number of people with the hyper-CVAD, couldn't collect enough stem cells. We said, that's not good. That arm was closed. The bendamustine rituximab transplant arm stayed open until about 40 patients, but now it wasn't going to answer a critical question, so we got enough experience to say, yes, you can do this, yes, you can collect stem cells. And the early results look promising, but it's no longer a randomized trial, so you don't know if it's just patient selection. So the system works to protect patients who go on trials. It's not just, well, we'll wait 5 years. It's active monitoring to make sure that we're not causing issues. So those were the 2 U.S. international group trials. This is called the triangle. This is a slide courtesy of Martin Drayling, who really runs the mantle cell lymphoma in Europe, out of Germany. And if you read any papers, he's on anything out of Europe and a lot of the U.S. stuff. And it's a 3-arm study, so you get randomized to one arm, A, B, or C. And so what they had done over the years, they had done a randomized trial with R-CHOP transplant or R-CHOP alternating with R-DHAP. This is high-dose Ara-C, platinum, and dexamethasone, a steroid. It's a standard salvage regimen, second-line regimen in lymphoma. They moved it up front. And they found that the alternating this was better than just giving R-CHOP, followed by the transplant. So that's now their new standard without any maintenance. Ibrutinib comes along. We want to move it into front line. It should work well. So the next question is, if I give ibrutinib with all of this, is that going to be good? And they decided to give 2 years of ibrutinib maintenance rather than forever. So they're going to test the question about, can you stop after intensive therapy and 2 years of ibrutinib? And then they ask a question which we don't think we can ask in this country, which is chemotherapy with ibrutinib, 2 years of ibrutinib, observation, no stem cell transplant. Now, in Germany and Europe, patients seem to be more willing to accept randomized trials with sort of a big difference between arms. In this country, not so much. And doctors, a lot of doctors are biased to transplant, wouldn't enroll. So we didn't think we could do this trial in the U.S. But they're doing it, and I think it's a very important trial. So it's asking two questions. One, in the era of new agent ibrutinib, do you really need a stem cell transplant? The number of people with MRD after R-CHOP, R-DHAP by itself is in the 80% range. Plus ibrutinib, maybe to be higher. So that's an important question. And there's 2 years of ibrutinib maintenance versus nothing. Now, it's not rituximab, so, again, at the end, we'll be scratching our heads a little bit, but it's a good trial. So this is open, accruing. We're going to wait and see what happens. This trial has finished accruing, and we're, I think, expecting to see the early results at the American Society of Hematology meeting in December. Can't promise you that. And it was similar, saying, okay, age over 65, most of it's BR, followed by rituximab for 2 years. What if we add ibrutinib through that? Okay, I added in induction, I added in maintenance, and then I continue it until the disease comes back. So that's fine. The end point is, how long does the disease stay away? Perfectly valid design. The problem is, this should be better, right? You're adding a new drug. It can't, you don't think it's going to be worse. It could be, it could be toxicity, but you think this is going to be better. But the big question is, what if I got this and got, 3, 4, 5 years before I relapsed and then started ibrutinib at year 4 or 5 when my disease came back? Is that a better overall strategy to keep the disease in remission than using all of it at frontline? So I'll have a longer disease remission with the extra drug, but have I really helped you in the long run? And that's a question that we don't always get out of clinical trials. Once we stop the trial at progression, we really don't follow patients to see what happened. So that's an interesting trial, and we expect to have some results in the next couple of months. So this is sort of my thought of, that what I was just talking about. Like, if you start the drug here, if this is bendamustine rituximab, the disease goes into remission and then it starts to grow back. If we do sensitive techniques, we might detect molecularly the disease back here, and eventually the nodes will be back here. If I add the new drug here, it'll look good. My time to progression will be better. But what if I added it here as a consolidation? What if I added it here when the disease started to come back? What if I just wait till it comes back and treat it? You might be in the same place. And that's the question that we don't know. And we have to keep in the back of our heads when we look at these trials. All right, so let's talk about novel strategies. So this is a different way of looking at young fit. You get intense therapy. The idea of the Germans was to add ibrutinib, so biologic therapy. Older, less fit, we're looking at BR, maybe with Velcade. And then we're looking at a biologic combination, maybe Revlimid rituximab, which has early data that looks promising. And then maybe add a biologic to it. But again, we didn't think we could ask transplant versus not. So the current trial that's just opened recently in the U.S. through the Eastern Cooperative Oncology Group is this. Doesn't matter what your doctor decides to treat you with. It'll probably be R-CHOP, DHAP, Nordic, whatever. Could be BR, doesn't really matter. At the end of that induction, you're finished with your 4, 6, 8 cycles of treatment. We do the MRD test If it's negative, then we say, you know, you've had a great response. Now maybe we could ask the question of stem cell transplant or not. So if your disease is still there molecularly, you get the transplant that your doctor planned originally. But if you're negative, which is a fairly high percentage of patients, then you agree to be randomized to get a stem cell transplant or not. Now, most people will have their stem cells collected and stuck in the freezer, so it's really not stem cell transplant or not. It's really stem cell transplant now or eligibility for stem cell transplant later, maybe a few years from now. So that's sort of the design. So this study is open, and if you're on treatment, first-line treatment, you know, your doctor could say, hey, you know, 3 months from now when we're done, we'll talk to you about this trial. So the big questions are really, does adding ibrutinib or other agents to intensive regimens improve outcomes? That might be ibrutinib, that might be lenalidomide, other proteasome inhibitors, et cetera. Does adding the agent to non-intensive, like bendamustine or rituximab, improve outcomes? And that might be the SHINE trial, BR ibrutinib. There's another BTK inhibitor, cousin of ibrutinib, called acalabrutinib, and there's a trial ongoing looking at BR plus that. And lenalidomide, you know, the R squared looks good. Maybe we can add lenalidomide in here like we do in the ECOG trial. We're adding it as maintenance, so we'll see how that does. And ultimately, can we get rid of the bendamustine or other chemo regimens? And that's that R squared, the lenalidomide rituximab. You could use the rituximab or some other anti-CD20. There's lots of trials like that. Ibrutinib plus rituximab looks pretty active. I mentioned the BR plus BTK. That's not replacing it. And at the Cleveland Clinic before I left, we opened a trial of R squared plus the proteasome inhibitor ixazomib, which is oral. Velcade subcutaneous or IV. This is an oral agent. So again, you can think of a zillion combinations. We just have to be strategic. But those are sort of what I think of the big outlines. Yeah, so that one is an frontline, you've never been treated trial. But we, you know, and people are looking at it. There was a large trial in follicular lymphoma, other indolent lymphomas of R squared versus R chemo that we are waiting for the results of. That, I think, you know, would make us even more excited about doing it in mantle cell. But again, there's some resistance to doing an R squared versus R chemo trial. Either I want this arm or I don't want this arm. So we've talked about such a trial, but a straight-up randomized phase 3 is not, to my knowledge, open looking at that in mantle cell in particular. So it would be a selected phase 2 looking at R squared plus something, or, you know, some variation of that. I think also that the ECOG trial, where we did the R squared maintenance, may tell us if that really is great, it'll say, gee, do we even need the induction treatment? So there's signals that'll come out over the next year or two that might make us more or less interested in using, you know, in studying R squared itself. So usually what happens, so, you know, you talk to that study, the R squared study that Cornell did. It's published in the New England Journal of Medicine. Very promising results, about 30 some odd patients. So you heard this morning at the session that one of those patients was on that trial. But, you know, he said he was a watch and wait, 3 years, and then went on the study. So is he typical? How many of those 30 patients were high MIPI risk, low MIPI risk? How many were sort of more indolent? So it's very exciting data. We like it, you know, in terms of consolidation maintenance, but it doesn't mean that should change the standard for everybody else. Other people are doing additional studies on that, so we'll have larger numbers of patients treated. We'll see if it holds up. So so now we need a larger phase 2, maybe multiple institutions. A lot of times when things go a single institution with selected patients to larger numbers of institutions and wider patient populations, the results aren't as good because the patients are sicker and, you know, have harder disease to treat. So, you know, a 30-patient one's we're all encouraged by that. I don't want to poo-poo the idea. So R squared is a very exciting regimen, but it's not necessarily ready to replace some of these other ones.
Why do you think it hasn't happened yet for those patients?
Oh, it takes time to open these trials. There are other trials, R squared plus. So people are building on it already. They say, gee, that's good. Let's R squared plus ibrutinib, R squared plus proteasome inhibitor. So it's being studied, and I'll show you some of those in a minute. Yeah, no. So the data in the R squared is studied, but again, it's 30 some odd patients. You don't know how many people were observed for a period of time before they got there. I don't think, if I remember, they didn't have Ki-67. I'm not sure about that. So yes, they have some data. It looks very promising. I mean, we're all excited about that data, and we're building on it. But it's just, it's not like, we're going to throw out everything else, and that's going to be the study. The ideal study is exactly, R squared or R squared plus something versus R chemo, randomized phase 3 would be the coin of the realm to say we've really made it in advance. Absolutely. So you really want to see that in the study, but it's what happens if you have 30 patients. Now you've got 8 with high and 12 with intermediate, and so the statistical power gets lower, and that's the problem. We really need more patients to really sort that out. Yeah. So we're all excited about ibrutinib. I think, where are we going? So I looked up clinicaltrials.gov, which I highly recommend. You can put in mantle cell lymphoma, you can put in frontline, relapse refractory, whatever. You can put in your drug of interest. And I think if you looked this morning, you'd probably get, if you looked at U.S. mantle cell interventional trials, you'd probably get about 100 trials open, so there's no shortage of trials. Then you have to pare it down. How many are frontline relapse? How many are stem cell transplant versus not? But there is this trial I mentioned a few minutes ago, mantle cell lymphoma, age over 65, bendamustine rituximab, with or without acalabrutinib. It's ibrutinib. It inhibits the same enzyme. It appears to be a little more focused on the BTK and less what we call off-target effects, where it hits other enzymes which are similar. And it's very similar in design to the SHINE trial. Endpoint is how long it takes the disease to progress. 500 some odd patients. It's a big trial, company run. Company has a very vested interest in showing that this is, a good drug. The problem I have is I assume it's going to be a positive trial. So now we know BR plus ibrutinib. I'm assuming the SHINE trial will be positive. This will be positive. And does that tell us whether ibrutinib or acalabrutinib is better? And that's what we don't know. And does anyone really want to do 500 patients to see which of those might be a little bit better? I mean, or can we ask better questions? If we have 500 mantle cell lymphoma patients willing to go on trials, what's our best use of those patients? So that's something that's going to come up. We're very excited. We and others have, got this idea that B-cell signaling is one pathway, B-cell death is the other. That's a logical thing to combine. Out in Cleveland, in Eric C's lab, he gave the pathology lecture yesterday. We did combos trials and clearly showed benefit of ibrutinib venetoclax. Other groups have done the same thing, and that's in clinical trials already. So I think that's a very nice combination. They're both oral agents. They're both pretty well tolerated. So that, and then if you throw in an anti-CD20, there's a pretty advanced trial in chronic lymphocytic leukemia, ibrutinib venetoclax, and a CD20. So I think that's an area that we think is very exciting, potentially practice-changing, and in the long run, the kind of thing that we think may lead to long-term remissions.
Both first line or?
So the acalabrutinib study is first line. The ibrutinib venetoclax studies are in relapsed. Yep. If they look promising, that would be an obvious combination then to move up front to front line. Can we get rid of chemotherapy?
What is BTKI?
Oh, I'm sorry. Bruton's tyrosine kinase is the enzyme, and the little I means inhibitor. So Bruton's tyrosine kinase inhibitor. So Bruton's tyrosine kinase is a key enzyme in normal B-cell development, and that's what ibrutinib targets. And that was hidden away in Dr. C's slides yesterday, but yeah, we usually get lost. So it's really a fascinating story. So Dr. Bruton was a physician back in the 50s, and he noted that there were a bunch of boys who had had repeated infections and no antibodies. At that time, we didn't even know about B cells. Later, it became clear that they had no B cells. And in the early 90s, they cloned the gene that was in those kids. It's on the X chromosome, which is why boys get it and girls don't. And it was an enzyme, and then it was really a targeted effort to synthesize an inhibitor, which turned into ibrutinib, which turned rapidly into clinical trials, and got it's really a success story of targeted therapy. So it's Bruton's tyrosine kinase to honor his seminal contribution to the disease, and the inhibitors. Yeah, in the back.
So maybe I'm jumping the gun on your next slide, but I see that both of these are for patients over 65.
Right. So because most people still go with the more intensive therapy for patients under 65.
But you relapse.
Well, so that's the bottom line. The older one, yeah, I think that's a little bit, you could argue that should be open to everyone. And there probably are such studies open to everyone, but this is a phase 3 randomized trial. They're trying to show that their combination is better than ibrutinib. And I think they want to focus it on a particular population. Now, since the average age of diagnosis of mantle cell lymphoma is in the 60s, and this is relapsed hopefully several years after they're diagnosed, it actually captures many patients with mantle cell lymphoma. Maybe not everyone in the room, I see, but but that's the So when a drug company is looking to a trial called a registrational trial, they are very careful about defining the patient population, because that is going to translate into the labeled approval that the FDA goes for. And so those trials, if they just want to find out how well something works, they're usually a little more generous. But if they say this is what's called a registrational trial, we've talked to the FDA, if our trial is positive, we will get approved, or they'll strongly consider us for approving for this indication, then they are usually pretty strict about the characteristics, because they don't want to mess up a chance to get approved. There's a lot of ibrutinib is a great drug, but as we talked about, do we really want to continue it for the rest of your life? Ideally, yes, but maybe we don't have to. We don't know the long-term toxicities. It's been around for 4 years. We already heard earlier, and we see hypogammaglobulinemia. You don't make antibodies, you're prone to infection. Now I got to replace IVIG, which is expensive, has its own issues. There is some increased risk of bleeding when you're on ibrutinib. It's difficult to manage anticoagulation if you need it, say, for atrial fibrillation, which happens to be a side effect of ibrutinib also. So how we manage the bleeding risk can be a problem. I'll skip the rituximab because that's just theoretical. It actually hasn't been a problem. We don't know, as I mentioned, the best way to use it, starting with the drug. You could, in theory, if ibrutinib blocks a cell from dividing, it might not be good to give chemotherapy at the same time as you give ibrutinib. It might protect the cells. We don't know. Just theoretical things we need to think about. And there is this literature about at least patients who went on early who had a heavily pretreated disease, when they progressed after ibrutinib, had very aggressive disease, almost blastoid-like mantle cell. And so that's a concern. Where we're selecting for bad actors. Seems less of a concern when you get it early in the course, but again, it's in the background. So, it's not a perfect drug. No drug is. We already know that the cancer cell can become resistant. The most direct way is to just mutate the binding site in the BTK, so now the ibrutinib has no place to bind. There's an enzyme just downstream, so if you want to say this is blocking this signal pathway at BTK, the cell says, well, I'll just bypass that. I'll have a mutation that activates the gene downstream, and I don't care. You can block BTK all you want. You can come around and bypass the dam with the PI3 kinase, and there's drugs like idelalisib which block that, so that's a rational combination. And there's mutations in the adhesion molecules that keep the mantle cells or lymphoma cells attached to the bone marrow of the lymph node that affect how well ibrutinib works. So again, we have to, when we have a new drug, we immediately start thinking, how is that cancer cell going to figure out a way around it, and how are we going to block that and make it think again? So again, clinicaltrials.gov, ibrutinib plus. These are all trials that are open on clinicaltrials.gov. I didn't make them up. They're open and accruing in various sites. So ibrutinib plus bortezomib. I didn't find with carfilzomib, which is a bortezomib-like drug, but I think there's one around. That's based on some science in the laboratory that said if you screen different pathways, BTK pathway and the proteasome pathway seem to work really well together. You can add ibrutinib, rituximab, ublituximab, all these CD20s that are out there. Venetoclax we talked about, venetoclax plus a CD20 we talked about. Lenalidomide, so lenalidomide, rituximab plus ibrutinib. It's a way to build on the results of the R-squared, maybe make it better, don't know. We're going to talk about immunotherapy, but the checkpoint inhibitors you heard about, I think after lunch the other day, ibrutinib has some effects on T cells and actually the immune system. It's not just directly on the cells, and we think we can take advantage of that by adding these cells which make the T cells work better. And then there's this company that makes ublituximab and this PI3 kinase delta inhibitor. Now, idelalisib, which is approved in CLL and other diseases, Brad Call, who talked yesterday, ran a trial in Mantle cell. The cells responded, but they rapidly became resistant. So the duration of response was too short, so we kind of said, not so good. There's another PI3 kinase just approved last week, I think, or in the last month, called copanlisib, not for mantle cell, for other indolent lymphomas. Maybe we have to rethink that. And this drug has a little bit different specificity pattern. Maybe we can get less toxicity and more efficacy. So PI3 kinase delta I showed you in the previous slide is a way around BTK. So again, there may be areas here, but no shortage of trials. If you tell me which one's the best one, I don't know. That's why we do the trials. I may have a bias. I might be totally wrong. Now there's practical aspects. Venetoclax is oral. Ibrutinib is oral. Some of these drugs are IV. How many times do I have to go to the site? I live 3 hours away. It costs $40 to park in New York. How many extra visits? This trial might take one extra visit a month. This might take 3 days in a row. So sometimes practical things tell you which is the best trial for you rather than the science. This is a cautionary tale. Entosplitinib was that Syk inhibitor. We said, let's add it to idelalisib. Rational combination. Phase 1-2 trial. Phase 1 part, because we're taking two drugs that we know how they work, we know the dose. We cut the dose a little bit. We say these are two active drugs. This is one of those phase 1s that should work, and we'll rapidly go up to the full dose and see if it works. There was unexpectedly inflammation of the lung. Trials monitored closely. After a few cases, the red light went on, said, this is not good, let's stop the trial. And so that combination is no longer being explored. So, yes, people on that trial took some risk. They had some side effects. Doesn't mean the disease didn't respond, but they had some unexpected side effects. These things are monitored closely. Trials shut down, and we go, okay, let's move on to the next thing. So there are cautions. Not every clinical trial is a success. This is the palbociclib I mentioned. I'm not going to go into detail, but cyclin D1 and these cyclin-dependent kinases drive the cell through the cell cycle. And again, we did this trial on 17 patients, and we asked them to undergo a biopsy at the start and a biopsy 3 weeks on treatment so that we could see whether this stuff actually did in the patient what it was supposed to do based on the earlier experiments. And forget about the sign for a second, but this is the phospho-RB, which is a sign that the cell is dividing. You can see that we dropped it. And the Ki-67, 3 weeks into treatment, also dropped by a half. This is a FDG PET, the standard glucose PET scan. 3 weeks later, absolutely no difference because the cells are not dividing, but they're still taking up sugar. However, this FLT, this is thymidine, which is a precursor DNA. Experimental way to do a PET, we label it, we give it to the patient. And FLT PET, it's halfway down. So DNA, the cells were not dividing. They were dividing about half the rate as shown here by an experimental test, but the FDG PET early wasn't so good. Later on, it would become good, but so again, all these fancy endpoints that we do to try to prove that we're doing what we want to do. So again, a list of most of the things we talked about. The couple things I want to spend the last minute or two on before opening to all questions would be the immune therapy, the CAR T, and the nivolumab pembro a little bit more, and the bispecifics. So this is adapted from a review in Blood. So we want the tumor cell, in this case mantle cell lymphoma, and we want the immune system to kill it. How is it going to do that? Well, mostly it's T cells and natural killer cells. And we want to get that T cell next to the lymphoma cell. And we can do that in a number of ways. So we can say, yeah, there are some T cells that bind to the lymphoma cell. They're just not active enough at killing. People call them exhausted T cells. But these antibodies, these checkpoint blockades, so pembrolizumab, nivolumab, you can't turn on the TV now without Opdivo for lung cancer. These are widely active drugs because they activate the T cells to kill whatever they're designed to kill. And they do work in mantle cell lymphoma. They're not a home run, but they're there, and we're trying to figure out how to use them better. But it takes the T cells that are already primed to kill the mantle cells and makes them work better. We can say, gee, there's T cells floating around, but they don't find the mantle cell. So bispecific antibodies, this drug blinatumomab was recently approved for acute lymphocytic leukemia of childhood. It has an antibody, two antibody ends, so that's what we call bispecific. One binds to CD20, and one binds to CD3, which is on T cells. So if it finds one cell and then it's floating around, it brings the two together. So it doesn't make the T cell kill, but it just brings it next to the CD20 positive cell, and now it kills it. So that's a very active drug in ALL, being studied intensively in B-cell lymphoma. And if you're an engineer, you can think, I can design a bispecific with all sorts of things, and it's just the sky's the limit. And then finally, this week we got the CAR T-cell approval in diffuse large B-cell. It was approved, different CAR T product for ALL. So CAR T is definitely on the horizon. There is a trial, there are several trials accruing mantle cell lymphoma patients for CAR T. One of my patients was treated in Cleveland in January on one of those trials. And so, that's something that people are pretty excited. You heard this morning, if you were there, about a woman who's basically alive for CAR T for diffuse large B-cell who probably wouldn't be here otherwise. So we're all excited about CAR T cells. It's not for everyone. People get very sick or can get very sick in the week or so after. So you have to be in pretty decent shape. And there's lots of caveats, but it is exciting. So immune therapy is sort of where we're at. And then just to say CAR T cells in diffuse large B-cell lymphoma, probably right now, we're thinking in the 30 to 40% range in terms of patients who we think are cured or at least have durable remissions. So it's not perfect, but already CAR T, ibrutinib affects T cell function. I told you that. So CAR T cell with ibrutinib. Interesting that patient we heard from this morning had ibrutinib to bridge her to transplant. Maybe, unbeknownst to anyone, that had some benefit in making her T cells work better. We had a patient who's going to be presented at the ASH meeting in December who got CAR T cells for large B-cell lymphoma, and 2 weeks later, disease was growing right through. All right, failure, doesn't work in everyone. What do we got to give him? We gave him nivolumab, and he had a spectacular response. So he could have been responding to nivolumab, but we actually have showing that his CAR T cells re-expanded. So we reactivated these CAR T cells that by themselves were not able to kill the lymphoma. We reactivate them, and they do. And Steve Schuster at Penn has a case like that and is a trial looking at that. So that's one way. Lenalidomide, immune modulating. Maybe we can mix that with CAR T. Some people, at least in the ALL, the kids with ALL, become resistant to their CAR T because they get rid of CD20. Cancer cells are pretty smart. And so now people are looking, well, can I mix a CD20 and a CD22 CAR together? And it's less likely the cancer cell will be able to downregulate both of those. And so again, we just have to always stay ahead. Why didn't it work? Why didn't it work forever? How can we make it better? And if I've left you with anything, there is no shortage of clinical trials. What there is starting to be is a shortage of patients willing to go on clinical trials. Gina Colada, who's the science writer for the New York Times, had this big headline on the front page: too many drug trials, too few patients. And I think we have to encourage people to go on trials. We have to be strategic about which trials we open and what we do with the data and interpretation. So with that, I'm happy to take questions. Didn't leave as much time as I wanted, but we answered some along the way, so yeah.